Hello friends, welcome back to my channel. Today I will start Krebs cycle. That is very interesting. So, first introductions of some Krebs cycle. Krebs cycle is common metabolic pathway which occurs in all type of body cells, especially those which are active cell requires energy which they get from Krebs cycle and etc. So, I will start Krebs cycle procedure. Krebs cycle, the first step of Krebs cycle. So first I have presented pyruvate structure, that is easy structures. So before Krebs cycle, the pyruvate present in the cytoplasm, the carried into the mitochondria with the help of carried. So this pyruvate is converted acetyl CoA, the help of enzyme named pyruvate dehydrogenase enzyme. SCOase enters in these reactions and CO2 eliminate from pyruvate and enter NAD plus and produce NADH plus H plus. So form of acetyl CoA. The second step, the first structures of acetyl CoA condense with oxaloacetate. So I have presented acetyl CoA structure and oxaloacetate structure and producing the form of citral CoA. Citral CoA. This reaction is very important. Formations to citral CoA, which undergo further hydrolysis with the help of citrate synthesis. So citral CoA, which undergo further hydrolysis with the help of citrate synthesis. So citral CoA HOH combined these reactions, but that is not to the shown mandatory. HS CoA eliminate from citral CoA and from citrate. So as citric acid is the first from compound of the Krebs cycle. Therefore, this cycle is also known as citric acid cycle. Moreover, since citric acid contains 3 carbon containing group, therefore the name of this cycle also known as dicarboxylic acid. So the third step, similarly, I have presented citrate structure. Citrate from now undergo the reactions in which water molecule is eliminated from the citrate. The enzyme helps these reactions aconitase and the formations of cis aconitase when H and OH and water molecule eliminated from the citrate and formation of cis aconitate. This process also known as dehydration process. Step 4. I draw cis aconitase structure and cis aconitase structure. So in these reactions, enzyme is aconitase enzyme continue the further reactions in which a water molecule introduced into the double bond in the opposite directions, which is removed. Isocitrate is produced, which is actually similarly to the original citrate molecule. Only the OH group is the differently positions and the formation of isocitrate. The fifth step, I draw isocitrate structure. In these reactions, isocitrate is converted to alpha ketoglutarate. So first, I say in these reactions help of enzyme isocitrate dehydrogenase. This enzyme which not only oxidize isocitrate but also decarboxylated it in these reactions. NAD plus enters and produce NADH plus H plus. And formations of oxalosuccinate, that is very much easy structures. Oxalosuccinate structure, and this is the first oxidation steps of the Krebs cycle. Due to the oxidation, oxalosuccinate is first produced which undergoes spontaneous decarboxylations, forming alpha ketoglutarate. Of structures, alpha ketoglutarate, that is the easy structures, alpha ketoglutarate. And next, this reactions helps to maintain enzyme alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase in these reactions enters H-square, enters NAD plus, produce NADH plus H plus, eliminated CO2 eliminated CO2 from alpha ketoglutarate and formation of succinyl CoA. These reactions, please remember that similarly to the first step of Krebs cycle. When I draw succinyl CoA is changed to succinate. The enzyme succinate thiokinase. In these reactions, AHOH enters and eliminate H square. Eliminate the H square and enzyme which breaks the bond, and the energy release is enters within a GTP molecule. GTP is converted ATP, itself becomes GTP by the enzyme nucleotide diphosphate kinase. Step 8. Drop structure succinate. Succinate is converted fumarate that helps to enzyme succinate dehydrogenase enzyme. Succinate dehydrogenase enzyme and enters in this reaction FAD and produce FADH2 and formations of fumarate. FAD is a coenzyme. I have presented fumarate structure, that is fumarate structures is converted malate. Fumarate is hydrolyzed to produce malate, by which introductions of water molecule this necessary. These reactions and water molecule break the double bond. Usually the malate is produced, the enzyme fumarate and product from malate. And now malate structures. Now malate is oxidized by malate dehydrogenase enzyme. So the enters NAD plus and produce NADH plus H plus and formations of oxaloacetate. The coenzyme area used here is NAD plus, which remove hydrogen ion from malate with the formations of oxaloacetate. After complete of one cycle, this oxaloacetate can now pick up another acetyl CoA and start TCA cycle.